Good afternoon and welcome to this webinar on student research in biology at Luther College. My name is Kirk Larson and I am the chair and professor of biology here at Luther. And today we are going to be joined by two of our current biology students, Moretta Hansen and Lena Schmidt, who will both be giving short presentations about their research in quite divergent areas of biology. A bit of information on how we plan to structure this webinar, and somebody just want to give me confirmation that you are hearing me okay. Okay, thanks. Following each of our student presentations, we will have a real short question and answer time about their specific presentation, just on the content of the presentation. And then we're going to follow up both of their presentations with kind of a more general Q&A session. Where you can ask some general questions. I'm actually going to start that session with a few questions for our presenters, and then we'll open up the floor for additional questions from the audience that you can address either to students or a number of biology faculty who happen to be present. Now, during the question and answer sessions, you can either raise your hand, if you've figured out how to do that, hopefully, or you can use the chat feature and type in your question, and we'll try to get to as many of the questions as we can. And we'll see what general themes kind of come up. Now, one request, please keep your audio muted unless you're called upon to ask a question, and feel free to keep your video muted if you'd prefer that throughout the webinar. Additionally, I'll mention that this presentation is being recorded for viewing by students who are unable to join us today. I'd recommend using presenter view so that you will be able to see the slides that our presenters are presenting during their talks. We hope this is the first in a series of webinars with the Luther Biology Department over the next few weeks that will allow students interested in biology and looking at college an opportunity to find out what it's like being a biology student here at Luther College. So once again, welcome to Luther College. And at this time, I'm going to invite Moretta Hansen to introduce herself and talk to you about the research that she's been performing.
Hi, everyone. My name is Moretta Hansen. I'm a senior here at Luther and a neuroscience major. And what I'll be presenting for you guys today is actually a poster that I'll be presenting next week at a genetics conference. So this is one of the earlier run-throughs of it. But my research is focused on neuroscience and genetics. I work with Dr. Fredham in a neuroscience lab, and we're very interested in this signaling pathway called the PI3K-AKT pathway. So this pathway here that you can see is actually a subset of the entire insulin signaling pathway. Now, insulin signaling is critical for a lot of different things, especially cell growth. So this PI3K-AKT pathway has been looked at in relation to cancer a lot, because if it's overly active, you get cells that don't stop growing when they should. But it hasn't really been looked at in relation to other diseases, such as neurodegenerative diseases like Parkinson's or Alzheimer's. So that's kind of what we're focusing on here, is how does either an excess or other problems in this signaling pathway contribute to issues in dopamine function. So to look at this, we're using little nematode worms called C. elegans. So these worms are just a couple of millimeters long. They're very, very small, but they grow quickly and reproduce quickly as well, so they're really great for research. A lot of labs around the world use them. But what we're using is an N2 strain, so this is just your normal C. elegans. But what else is nice about these is you can actually manipulate their genetics so that you can force some of these animals to have issues in this insulin signaling pathway. So some of the strains that we work with have either excess signaling or a lack of signaling, and then another strain that we work with a lot has issues in its dopamine function. So using all of these various strains, we can then run a couple of different experiments to see how these animals respond. Do these ones with divergent signaling in the insulin pathway respond more similarly to our wild type or normal animals, normal worms, or do they respond more similarly to the ones with problems in dopamine? So the first experiment that we did is what's called tap habituation. So habituation is learning to stop responding to a stimulus. So if you think about it, if somebody taps you with the same amount of force over the same amount of time repeatedly, at first you would respond, and you'd respond quite a bit, but then as you got used to it, you'd stop responding. So this is habituation. And so I actually was able to build a Lego robot that taps the plate that these worms live on to see whether they stop responding or not. And so that's what you have over in figure 1 and 2. These graphs show the results of this experiment. So over a number of taps, the worms do stop responding the way that we'd expect them to. Now, purple is the normal worms, and blue is ones with issues in dopamine. Dopamine is a neurotransmitter that's critical for movement and learning. So learning to stop moving is all about dopamine. And so we see these animals that have issues in their dopamine function do have a different response to this tap habituation like we'd expect. But then down in figure 2, this is where we compare ones with different insulin signaling to see if they respond similarly to our normal ones or to the ones with problems in dopamine. So these animals here in orange, they respond pretty similarly, but actually they respond a little bit better than our normal animals. And so these are ones with a lack of insulin signaling. They don't have as much signaling as normal ones. But these ones in red, this is what we're interested in. So these worms here, they have a mutation that causes excess insulin signaling. And so their response is a lot more similar to these other animals up here with problems in their dopamine function, rather than it is similar to wild type or normal animals. So that's the first experiment we did. The second one is induced fluorescence. So under certain conditions, we can actually cause dopaminergic neurons to fluoresce. And so doing this allows us to see, well, do these animals, these different strains, have the dopaminergic neurons we'd expect them to? So wild type animals, they do have these neurons as we would expect. These age 1, the ones that lack insulin signaling, they also have dopaminergic neurons like we'd expect. And over here is where it gets interesting again. So these animals here, these are the ones with problems in dopamine function, and they actually don't show these neurons like we would expect them to if they were normal. And these DAF18, these ones with excess insulin signaling, they also don't show neurons like we would expect them to. So again, these animals are a lot more similar to ones with problems in dopamine signaling than to our wild type animals. And so our final experiment here, this again confirms kind of what we're thinking. This experiment used HPLC, which is high performance liquid chromatography. It's basically a way for us to measure the amount of dopamine that each strain has. So this here is wild type. These are our normal animals. So this is about baseline. And again, these animals with problems in their insulin signaling pathway or dysfunctions in this pathway show less dopamine. So overall, we can conclude from these 3 experiments that animals with excess signaling in the PI3K AKT pathway look a lot more similar to animals with problems in their dopamine signaling. So we do think that dysregulation of this pathway can cause problems in dopamine function in some way. And so in the future, our goal is to try to get at where that relationship is. So does excess signaling in the PI3K AKT pathway actually impact dopamine production, or does it impact the neurons themselves, or does it impact something else even? So thanks for listening to my presentation, and let me know if you have any questions.
So I think I haven't seen the result for the dopaminergic signaling before. It's pretty remarkable. So do you think that the neurons just aren't there, or are they just not producing dopamine? I guess I'm a little unclear in terms of what you're showing there.
Yeah, so that's actually one of the questions we have. Because it is dopamine-induced fluorescence, it could actually go either way.
Okay.
So using this, we can't definitively say whether it's one or the other. That would be a future direction.
Okay.
Well, thank you, Moretta.
Yeah, thank you.
Let's move to Lena. I'll unmute you and we'll put the spotlight on you. So,
All right. Hi guys, I'm Lena Schmidt. I've been doing research in the entomology lab for the last two years, since 2 summers ago with Dr. Larson. Let me share my screen here. And I have been doing research on moths. You guys all see this? Sweet. Technology is working. So our goal for this study, come on, is it gonna work? There we go. The objectives that we had to study the moths in the area were to compare abundance, species richness, and composition of moth communities in the planted tallgrass prairies and the oak hickory forests on Luther College campus. We also wanted to just find baseline, what moths do we even have? Because no one's ever really done a moth survey in this area before. So we studied 6 locations, 3 planted tallgrass prairies and 3 oak hickory forests. And I'll see if I can highlight. So our 3 forests were Hickory Ridge Woods, which is right close to campus, Spildy Woods, and Lineberger Preserve way out here. And then the 3 prairies were Anderson Prairie, Gateway Prairie, and Jewel Prairie. And all of this area enclosed in yellow is all Luther College land. So all of our sites got to be on campus, which was also very cool. So what we did in order to capture the moths is we used blacklight bucket traps, and these do exactly what they sound like. There's a black light in the middle, the moth is attracted to that, flies towards it, hits a plastic screen, and falls into the bucket. And we sampled over 5 dates in the summer of 2018, and we put these out 4 sites the first night and 2 sites the second, since we only had 4 bucket traps to work with. And we put them out in the evening before the sun went down and pick them up in the morning, and they'd be full of moths. So what that might look like is that top left photo is the inside of one of those bucket traps after being out for a night. So you can see there are tons of moths, but also lots of beetles, caddisflies, even some wasps and some other stuff. So the next job was to sort all of that out. So I spent a good amount of time sorting out moths from beetles, and then sorting all of those moths into species based on how they look. So all the similar moths would go into a pile, basically. And then we pinned and spread all of those moths. So we put them 2 to 3 representatives of each species on a spreading board, which helps to spread the wings out, and this makes it easier to identify the moths and also just to display them more nicely. And then I've spent the better part of the last year and a half identifying and labeling these moths, and a huge thank you to Abby Miller and Jim Durbin, who you can see in the pictures over there, for helping identifying some of the more difficult of the moths. So what we found, we collected a total of 12,290 moths, which is a lot. And we managed to identify about 80% of those, so 9,560. And most of those that we didn't identify was more because we ran out of time than because they were unidentifiable. A lot of micro moths left over, but what can you do? But of these 12,000 moths, or of the ones that we identified, they represented 34 different families and 468 different species, which again, a lot of moths. And 4 of those families were unique to the prairie, 15 were unique to the woods, and then 77 species of moth were only found in the prairies, unique to the prairies, and 248 moths were unique to the woods. So that was pretty cool to see. We also found quite a few state and county records. So state records would be moths that haven't been found and identified in the state of Iowa before, and county records having not been found or identified in the Winneshiek County. So we found 7 state records and 97 county records. And then comparing the prairies and the forest, so these graphs, the one on the left is going to be number of moths, and then the one on the right is species richness, or numbers of species of moths. And as you can see pretty clearly here, there are quite a few both moths and species of moths in the forests. So on average, we had 872 moths in the prairies, 3,224 moths in the woods, So that's about 4 times more moths in the woods than the prairies. And then on average, 129 species of moth in the prairie and 251 species of moth in the wood. So on average, about 2 times more species of moths in the forests. And then these two graphs show a similar thing, this time just splitting them up into the specific sites that they were in. So again, you can see quite a few more moths in the woods than the prairies, quite a few more species. But what you can also see here is that there was very similar numbers of moths and numbers of species within those habitats. So within the prairies, they all had very similar numbers of moths and numbers of species moths, the same thing for the woods. And this is also showcased pretty well in this next graph. So this is what's known as a principal components analysis. Basically what this does is it positions communities graphically based on similarities in the species that are there and also the abundance of those species. So the basic way to read this graph is the closer dots are together, the more similar their moth populations. So as you can see pretty well, the prairies are all clustered really closely together. That means there's not much variation in the moth populations between those 3 sites. So they had very similar types of moths and numbers of moths. You can also see then the forests are spread out a little bit more widely. So there's more variation amongst the moth species found in the woods. But also important to note is that the prairies over here and the woods over here are in 2 distinct separate areas of that graph. So that's showing that the prairies had very different moths from what the woods had. Then some of the more common moths we found. In the prairies, the most common was the pale lichen moth. There were 145 individuals of that species, followed by the bristly cutworm, the large ruby tiger moth, the confused dagger moth, and the harness tiger moth. So you can see these top 5 species range from 50 individuals to 145. And then you can see in the woods, the most common moth was the hickory leaf roller, with 401 individuals, then followed by the pink-barred pseudostrotia, the horned spanworm, the gray archips, and the zigzag herpetogramma. So you can already see just very clearly in the top 5 species that there are quite a few more in the forest, since the forest moths range anywhere from 253 to 401 in those top 5, whereas the prairies, again, the top one only had 145 individuals. And then, just for fun, some of my favorite moth species that we captured, the spotted apatelodes, the grapeleaf skeletonizer, Harris's 3-spot, the rose hooktip, and the luna moth. As you can see, they have some pretty fun names. And a lot of these guys were things I wasn't expecting to see, things I thought were really beautiful, some of the more rare moths. The spotted apatelodes, that was the only one of that species we found. So they were really cool to see. So in conclusion, again, 4 times more moths in the woods than the prairies, and 2 times more species of moths in the woods than the prairies. And we're kind of thinking that this might have something to do with the diversity of the plant life in these different habitats. But a little bit more research is going to have to happen before we can really say that for sure. And then the prairie moths were quite different from the forest moths, and you can always find a ton of moths and new species records when you look for them. So thank you guys for coming. Oh, acknowledgements, almost forgot. Probably shouldn't forget that. So a huge thank you to Abby Miller. She's a current sophomore at Luther. She helped out a ton with identification, labeling, and she's also organizing and databasing our entire moth collection. So huge thanks to her. And then Jim Durbin again, huge shout out to him for the help with identification. And also his website, Insects of Iowa, was huge just in the whole identification process itself. And then also a thank you to Luther College Dean's Office for providing funding for the summer research work that I do with Dr. Larson. All right, so again, thank you guys for coming. Do you have any questions for me?
Yeah, so Lena, thank you for the talk. I'm wondering, given I've read some papers about insect declines in many places, I think a couple studies in Europe, and I'm wondering if you have any sort of comparison controlling for sample intensity of the number of moths that you captured here compared to what other studies have seen. So just in terms of overall, not which species, but just sort of overall health of the population. Do you have any information on how what you're seeing compares elsewhere?
It's kind of hard to know, especially since none of these studies have really been done here. There was one sampling done in 1908 of Lepidoptera in general, but that is a really good question. I'm not sure.
Thanks.
Yeah. Other
Anybody-
questions?
All right, I'm gonna try and take the spotlight off of you, Lena. And let's see here.
Asking why did I focus on moths?
Uh-huh.
Yeah, I can answer that. That's well, I took entomology my sophomore year, and for that class we have to do an insect collection, and I love the moths. They're super fuzzy and adorable, and I think they're overlooked a lot of the time. Just everyone focuses on the butterflies, and nobody seems to care about the nice fluffy moths. So it was, I don't know. I just really like them. They're really interesting, and a lot of them are not just boring and brown, and a lot of them are gorgeous. So I just really like them.
Eric, you wanna ask your question there too? Did you have a second question?
Yeah, so my second question was the woodland versus prairie and whether that is a typical finding that grasslands in general have fewer species than, or again, that's just particular to Luther.
It's not what we were really expecting to see. From research, it sounds like prairies tend to have more moths in general, so it kind of surprised us. A lot of the diversity of moths is based off of the diversity of the plant life in the area, because most moths, as caterpillars, are specialists on a specific type of plant. So the more types of plants you have in general, the more moths you have. So the fact that we're not seeing as many moths in the prairies seems to suggest that the diversity of the plant life is less. I'd really like to do a study. There's a really nice older woman who lives a little bit off campus who has a planted prairie with like 200 something species of plants. And the ones at school have, I think, around 60 to 70 species that have been found there. I'd love to compare and see if that has an effect. But yeah, it was a little bit of a surprise seeing the prairies having that many fewer species.
So I'm gonna ask both Lena and Moretta. You can unmute yourself, Moretta. Just a couple questions here, and then we'll open it up kind of to general additional questions that people may have. But when you were a high school student, what was it that attracted you to Luther? And were you thinking about doing research when you were in high school at Luther or wherever you were looking at colleges? So either one of you can chime in right away.
I can start. So I've been wanting to do research since actually before I was in high school. And so that was one of the big things I looked for when I was looking at colleges. And what I found and what I'd been told mostly was if you go to a bigger college or a bigger university, you might have more variation in research, but you also run the risk of not having as much of a personal relationship with the people you're doing research for. So at Luther here, I work directly with Dr. Fredham in doing my research. And if I have a question, I can just run right up to her office, or I can shoot her a text, send her an email, and get a response very quickly. And I just love that more one-on-one, the close relationship that I get here.
Sorry, go ahead.
Go ahead, Lena.
Oh, I was going to say it was similar for me. My older brother actually went to school at Luther as well, and just the close relationship that the profs have with the students is really incredible. I was not thinking about independent research at all when I started school. I knew I wanted to do something with biology, but I sort of got surprised into entomology. Kind of wasn't what I was expecting to go into. So the research, actually Dr. Larson kind of really highly encouraged the research after I took entomology. So it was I think the closeness with the professors that got me into it. But also just the campus itself is gorgeous. There are these enormous gnarly trees and tons of natural areas and hiking paths that was a big draw for me.
All right, I'm going to ask you guys another question about how working on this research project has given you some additional opportunities for research at other places or here as well, and also what kind of experience or independent or internship experiences you may have had while you've been a student at Luther other than this research project here.
Yeah, so I spent my last summer at Johns Hopkins University, where I did an internship in a biophysics lab. And that was definitely influenced by my research experience here. They were looking for someone who had experience related to what I do. And really, it's opened up a lot of doors. So I'm a senior this year, and next year I'll actually be going to grad school at Ohio State University. And that's also because of the research experience I've had here, and being able to prove that I can do research and I can do it well is something that they're looking for, and something that Luther has helped me work on.
Yeah, I would definitely agree with that the research is super good at opening doors. If nothing else, it's something to put on a resume that looks really good. But I worked at the Minnesota Zoo last summer in their prairie butterfly conservation department. And I got to work hands-on with threatened and endangered species of prairie skipper, which is a butterfly. So for sure, having the research and again, having the close connections with the professors, I doubt I would have gotten that job if I hadn't had awesome recommendations from a couple of my professors. And it was an amazing experience. So yeah, it's definitely a door opener. I would highly recommend doing research. Even if you don't go to Luther, find somewhere to do research and do it, because it does open a lot of doors.
So I have another question about, have you had opportunities to present at professional scientific meetings or maybe other venues as a result of participating in your research? Moretta, you mentioned a genetics conference that you're going to be presenting at. What did you learn, or what kind of experiences have you gained by participating in these meetings?
Yeah, so like you said, I'll be presenting this poster actually at a genetics conference next week. I've presented other research at a nationwide conference in California. So that one had over 1,500 students that presented. And
what was the name of that conference?
That one's Abercombs, the annual biomedical research conference for minority students. And so that's every fall. But what I love about going to conferences is just the sharing of ideas. And so you can walk up to somebody and have no idea what their research is on, and just the passion that they share with you, and you walk away and you've always learned something, even if you didn't expect to. But that and the connections you can make just by going somewhere. So I also presented at a Midwestern conference, and I ended up talking to one of the graduate school heads at the University of Minnesota, one of their main recruiters. And so that was, I believe, part of why I got an interview there. And so it's just great for both sharing ideas and for connecting with others.
Yeah, I would, again, totally agree with that. I've gotten to present a couple times. I first presented last spring. It's been a while, at a conference called the Day of Insects, which was held at Raymond Gardens in Ames, Iowa. And that was more of a amateur entomology enthusiast meeting. So that was a really great opportunity to present on the first chunk of my research in more of a casual setting. And then last fall, I got to go down to St. Louis in Missouri and present a poster at the International Meeting of the Entomological Society of America, which was also really fabulous. And my poster actually won first place at the undergraduate poster competition there. So that was really exciting. And even better was the other girl who came with, Maggie. She works also in the entomology lab on ground beetles especially. Her poster won second place. So it was like, represent. But yeah, I totally agree. It gives you an option to connect with people and just to even see what are people doing in this field, what are their jobs, what are their interests, like where people are going with different things, and all of the new information that's being shared is incredible. Yeah.
So can you guys tell me a little bit about how you've been involved in research in some of the courses that you've taken? So not independent things in the summer or wherever, but in specific courses.
Yeah, so I've taken a couple of psychology courses as part of the neuroscience major, and I work with one of the psychology professors, Dr. Gould, as a kind of lab assistant / teaching assistant. And so for research in that class, I take out groups of students and we walk around campus and look for squirrels pretty much. And then for our research, we either walk directly at the squirrels or we walk at an angle, and we're either looking at the sky or we're looking right at them or we're looking at our phone, and we try to measure how long it takes them to respond to our presence. And so that's just kind of, it's a fun type of research that's pretty unrelated to the genetics neuroscience stuff I do, but it's also still relevant because we're seeing some interesting results between the squirrels that live on campus and other squirrels around the US that might respond differently.
Yeah, I had a ton of research experience in classes. I think pretty much every single wildlife bio course I've taken has had some sort of a small research component to it. Ecology, I was studying forbs and grasses in our prairies. In biostatistics, I think I was doing something with bacteria. That was a while ago. Entomology had research projects that you set up yourself and did the entire project and found the results. Actually, the couple of J terms I've taken have both had very small, since time was limited, but research projects. And I mean, yeah, studying a variety of things, plants and bees and fish and all kinds of stuff. So pretty much, yeah, and the environmental studies courses I've taken as well, soils, we were studying soil organisms. So a lot of opportunities in class to do research and to do it pretty much all yourself with the help of the professor. So doing all the research design yourself and then going out and collecting the data and then figuring out what it all means and writing up the paper itself.
All right, so let's open it up for general questions for both Lena and Moretta, or even just general questions about biology that maybe one of the faculty members need to answer. But Feel free to, once again, either type in your question using chat or raise your hand and we'll call on you and you can go ahead and ask those questions at this point. Thanks.
Hey guys.
Hey.
What's the average class size at your biology classroom?
Ooh. For my classes, I think the largest I ever had was 151 and 152. Those are the general bio. They've actually changed it since I took it. It used to be maybe 100, 150, but now they've done smaller class sizes. What is it, Dr. Larson? Maybe 30 per class?
30 to 45, depends on the year.
But that's about the biggest it gets. I've had classes where there are 7 students, and those are fabulous.
Mm-hmm. All right, thanks so much.
Yeah.
Hey, Jens, is your dad named Carl?
No, he's not.
Oh, okay, sorry. Just checking.
So I have a question in the chat then about neuroscience, and it's what drew me to Luther for a neuroscience degree. So when I started here, actually, Luther didn't have a neuroscience degree, so I was a bio and psych double major. And really, it was, again, the research experience. So Isabel, I don't know if you're looking to go into medicine or to go on to grad school or something like that, but Luther is pretty strong in both of those. So there's a couple faculty here. Dr. Carlson and Dr. Fredham are both on the neuroscience board or whatever it's called. And then Dr. Gould, who I also do research with, is on that as well. And really, it was the kind of experience I could get and the guidance I could get, because I'm the first one in my family and my extended family to try to go on to grad school, and having that personal relationship here was definitely helpful.
Dr. Carlson or Dr. Fredham, do either of you want to speak about the neuroscience program real quickly?
Trying to figure it out.
You got it.
Oh, am I there?
Yeah,
Hey.
you're there.
Okay, I should know how to work this by now. Hi, I'm Dr. Carlson, and I teach neuroscience and human physiology. Yeah, Moretta was one of the early students on in the major, and the major grew and kind of was developed in response to how many students we had going on to neuroscience graduate programs, and we've had a fairly strong history of that. And so Dr. Fredham, who is actually one of my former students and now is on faculty here, which means I'm old, is when she came, Dr. Gould, myself, and she, we all established this neuroscience program in response. And so we are now in our 3rd year of the neuroscience program. I'm very, very excited. It's been well received, and we're excited to see where our students are heading off. And now we have one of ours, again, heading off the graduate program at Ohio. And so we're thrilled with that. So if you have any questions about the neuroscience major, please feel free to ask.
So there's a question from Elise about studying abroad, and both Lena and Moretta, have either of you studied abroad? And can you talk a little bit about your experiences?
I can start with this one. I've had some really cool study abroad experiences. I did 2 with the bio department. So the first one would have been 2 Januaries ago. I did both J-term courses, so about the month of January was the length of the trips. But 2 years ago, I was in Arizona with Dr. Bach doing the Ecology of the Southwest trip, and that was incredible. We got to hike around the Grand Canyon and some areas in Colorado and learn all about the desert, and even do a quick research project in that course. And then this previous January, I was in Roatan with Dr. Larson on the marine biology trip, and that was hard to beat. There's nothing like scuba diving in the Caribbean for class. It's kind of hard to top that, but that was incredible, learning about the ecosystems of the coral reefs in that area, and all of the different fish and birds and everything. So yeah, incredible experiences.
So I've had a little bit of a different experience. I studied abroad for a semester. I spent a semester in South Korea, and so it wasn't specifically related to neuroscience, though I did take a neuroscience course at university while I was there. And really, I'd recommend studying abroad to anyone, whether it's a J-term course or a semester. I was able to fit it in with my schedule because I brought a couple of credits with me when I started at Luther. But I think it's a really invaluable experience, whether it's for research or research-based, or whether it's just to get out of the US for a little bit.
And Kirk, I might jump in there just to add to our students who are looking at this. Luther has a very, very vibrant study abroad program. It's amazing. And so like I took a group also to Roatan, Honduras to look at a different aspect, looking at poverty and ethical engagements there. And so there are opportunities throughout the US and throughout the world. And so it's a very, very exciting and very dynamic aspect of our education.
Mm-hmm. Now, Scott, maybe you can talk a little bit about what the biology type students or those kind of health did while they were on that trip that went with you, because they did some internships while they were there, correct?
Sure, yeah. My course was arranged that we had internships, and the students would spend the first 4 hours of the day working in these settings. We had 4 or 5 working in conservation, environmental conservation settings there. It's an interesting dynamic because there's literally no government enforcement or regulations, even though they do have laws. And so it requires these non-governmental organizations to monitor all of the environmental rules. And so they would be responsible to find violators and to drive the Navy out, which is just bizarre. But they worked with that. They worked looking with coral reef restoration. They looked at plastic pollution, an array of different environmental components. Another chunk of them worked in this clinic that is founded by a nurse from Ohio. And seeing how healthcare is delivered to a very impoverished region there. The average income is high for Honduras. It's about $15 a day. And versus the mainland Honduras is about $3 a day, but immense poverty. And so there's little health care. The government doesn't provide much. And so it's all done again by mission organizations and NGOs. And so the students had great experiences, spending half of each day working in these settings. Another group were in educational settings, working with the school systems there, which again are mostly non-governmental organization funded or missionary funded, because the average education is very, very low, 6th grade at best. 9th grade is the goal, but most don't do that. So that's an example of a different type of program that biology students might be involved with. And there are the ecosystem one. Kirk was right down the road from us, a couple miles away, looking at coral reef biology exclusively. So very exciting.
Yeah. Before I call on Enos, one quick question for you, Moretta, about your study in South Korea. I don't know if you saw that on the chat, but did the language barrier affect your learning at all?
Yeah, so fortunately it did not. So I studied at Yonsei University in South Korea, and it's a school of about 50,000 students. And because of that, and because of globalization, quite a few of their courses are taught in English. So most of the courses I took there were directed towards students that were studying there abroad, so those were in English. But then my neuroscience course was one for upper level neuroscience students that were looking to go to grad school in the US or in an English speaking country. So that one was actually taught in English to help these students in South Korea prepare. So it just worked out really well for me.
Okay, thanks. Dr. Enos, you have a question.
Yeah, I have just a quick comment and then a question for the students. Quick comment is an element that combines internships, study away, biology, neuroscience, and research that Luther is also doing that's might be good to be aware of is the Rochester semester. So it's a brand new program that I am co-leading and just started this spring. It's our closest study away, but the key driver of that, the reason that it's happening, is an immersion internship. So it is 20 hours per week on site in an internship, it's all majors, but we have 3 neuroscience and 2 biology majors up there currently in the inaugural semester, all in different research labs and there's some opportunity to have that be longer than a semester. Could stretch into a J term or in a summer. So very substantive experience that our students are having, and a lot of positive feedback from that. The question that I had for the students is, if you could look back and talk to your senior self, what advice would you give yourself that you maybe wish you would have known when you were a senior, when you were like in these choices and dilemmas and thinking about entering college, since that's where a lot of our audience is right now?
Ooh, I think the best advice I would give myself is that college is a holistic experience. So it's important to have the research experience that you want, but it's also important to go to a place that feels right to you. And what feels right depends on a lot of different factors. So do you walk on campus and are people generally looking happy? Are they excited to be doing what they're doing? Does it look like somewhere that you could be happy? Because I did definitely go looking for research, and I've been very fortunate in that it's worked out, but I think it really matters everything else as well.
Yeah, I guess I can't, yeah, that sums it up really nicely. I guess one more thing I'd add is look for, I mean, I personally really like the small school size, but look for somewhere that you will be comfortable, because some people do really well in the large school sizes, and that's a personal thing. Not every school is going to be good for every person, and vice versa. There are going to be some schools where, you know, you'll do just fine and you would have also done just fine somewhere else. Don't stress too much about making friends. I know it's a huge change in everybody's lives, and I was freaking out a little bit because I'm not the best at like initiating friendships. But if it's anything like Luther, they cram everybody together the first year in the dorms, in the classes. Here you have Paideia, where it's all freshmen, all the orientation, different activities. You meet people really fast, and I got lucky enough to meet some really good people. So don't worry too much about the social aspect. It'll happen.
So other questions from those of you who have joined us today? We'd be glad to try and answer those questions for you. So here's a question about online learning going, and how's the labs and research going during the quarantine?
It's not. I'm a little bit disappointed in how the semester's gone. So I was taking an anthropology course for fun called remote sensing, and one of the things we were going to get to do in that class is actually go to a site and like do the archaeology, you know, use the equipment and like find a foundation of a school or something. And we don't get to do that anymore. So it's a little disappointing. And working at home it's going.
Mm-hmm.
But the professors have been really good about taking input from the students and trying to make it as easy as possible.
Mm-hmm. Mm-hmm. In regards to your research, Lena, you've been able to at least work on your paper, which is a good thing, right?
I'm lucky enough that most of the in-lab research that needed to happen happened before I got off campus. But
Maretta, you want to address that?
Yeah, no, it's pretty similar for me. So I was able to get a bit of my research done over the fall and January. So I have enough with me that I can run my analysis on it from home and create my senior paper from that. Like Lena said, it's always better to be there and doing research all the time, but I'm very happy with the way things are going given the circumstances. So I'm in organic chemistry right now, and we're missing our labs for that class, obviously, but my professor's been going in and recording himself doing each step of the lab so we can see how it would be done. And so that's just really nice to be able to still kind of get the experience that we needed from this.
So any of the faculty want to talk about their online learning experiences so far, and especially related to labs, potentially? Something you've been able to come up with?
I'll share a little bit. And this would be for first year labs. So we have taken a strategy where we are also doing the videoing and we did our first YouTube video, a couple of us, and we did the whole lab. And then we labeled things and took students through step by step, and they can follow in their lab manual. And then what we did was took the previous results for this lab from a previous year and supplied that to the students so that they can get the actual data that would be coming out of that lab and can do the same analysis. And in some ways, there have been some real bonuses that were unanticipated. Like, for example, we can do a 2-hour lab. We're limited by that. Can't do replicates for some of the labs. All of a sudden, we can hand each group a set of 4 replicates of that experiment, and they can do a more robust analysis. Personally, I'm really enjoying the opportunity to Zoom with students, because we set up a lab session. We do breakout rooms, and I don't get to be in their room unless I go to it. And so I can visit their rooms, and it feels like I'm visiting their lab tables, and they're working in a group with their lab table, just like they would if they were sitting in the lab. So they miss everyone misses the hands-on piece of it. We're doing our best to make what we can happen, but still able to deliver a lot of the core learning objectives. And I mean, I know there are other faculty on the line here that are taking a variety of approaches, so they could chime in as well.
So in my upper division evolutionary biology course, we were able to complete most of independent projects in the first half of the semester. And we were sort of 1 week left in the lab before we started analyzing data. So the students told me what steps to carry out. And so I did that, reported back the results, asked if there were further steps they wanted me to do. Often there were, so we did those steps as well, sent them the results, and now they're working in their lab groups to analyze and pull together a poster. So it's really working, that aspect has worked pretty well. And this next project, we're inferring phylogenies using sequences from GenBank, and we don't need to be in a physical lab to do that. Any place we've got an internet connection can work, and so it's working out reasonably well. We got lucky on the timing.
Yeah, I'll just say that in my class, genetics, one of the ways we've tried to kind of supplement for the lack of being able to do hands-on lab activities is to actually read more primary research literature. So it's a kind of a big skill to try and develop over the course of your 4 years in college, is to actually read real research literature. And so I'm trying to introduce students to that and use that as a means of looking at, if we're not doing the research, how are other people doing that research, and how are they publishing it, and then what does that look like, and what are the best ways to go about understanding and really diving into that aspect of research science. So it's still kind of hands-off, but at least it's sort of looking at real data and really having a chance to look at the overall research process.
I'll say I'm teaching a course right now on insects, humans, and the environment for non-majors. And for lab, yesterday I was standing out in the middle of 10 Mile Creek with a tripod camera collecting insects, benthic macroinvertebrates, and going to be looking at the, I'm taking close-up images of all those organisms now, and the students are going to be doing some identification just like they would have been doing in the lab. They're also going to have to look at some of those shots and actually count the different kinds of insects in the bucket. They should be able to, the pictures I think are going to be good enough that they'll be able to identify them. So they're pretty much doing everything except getting their feet wet, unfortunately. But it really is a good, we've been able to do a few things, but from our standpoint, I think a lot of faculty would agree that this type of teaching is much more difficult. It takes a lot more work than doing it face-to-face, because you got to think through everything and all the possibilities and get everything posted online and get the videos shot, and it can be a lot more difficult. And then, of course, we miss having the students there when we're actually giving our lectures. It's getting the feedback of those students. So we're adapting to the situation right now. I wouldn't say any of us like it, but it's going okay. So, any other questions from our audience?
There's one on the chat. I can't really answer to that. I'm not in sports and my job is research. So
Okay.
I don't know if you have some experience with that.
Well, the workload of bio majors and sports or athletes or jobs or whatever. Good question. We have a large number of majors that are active either in athletics or in music. And both of those take a lot of time. And so every once in a while, there's some conflicts that come up, but generally, the students who are involved in athletics, the students who are involved in music tend to be pretty good about budgeting their time, getting things done that they need to do, sometimes better than students who aren't involved in athletics and music. So, but they definitely can do it. The workload for the major is going to be the same, or whatever you want to make of it. And so, Lena earlier mentioned the other student in my lab who did ground beetle research, and she actually was a two-sport athlete, which is pretty unusual, both soccer and basketball. And so she, of course, was very busy all the time with athletics, but she was doing research and she was doing very well in her classes as well. So it's definitely something that you can do. For pre-med students double majoring in a non-science area, does one of our kind of more pre-med advisor types want to take a stab at that one?
Yeah, sure. I'll take a stab at it. Yeah, it's very doable. We have quite a few students who do double majors, and we always say that it's a great thing. I mean, we always say don't do it just because you want to check a box for medical school, but I think there's a lot of value in doing double majors, and it's, if that's what your interest is, you can definitely do it.
At the same time, I would chime in here that I chase what you're interested in and let that be the driver, rather than feeling any sense of being compelled to take more, do double major, doing more minors, because what medical schools are interested in is diversity in the strongest sense, but listening to your interests as the driver. So if you really wanted to spend your time being a single major and really diving into X, whatever X is, that's the important thing, rather than, in other words, I wouldn't do things just to fulfill and check more boxes, but instead let yourself and your interest and your curiosity really be the driver, because that's what's going to come out authentically in a medical school application and in an interview.
And I'd chime in that when working, if you're in the medical field, you're working with human beings, which are more than just biology and chemistry. And there's an awful lot you learn about human beings in the courses outside of the sciences that are probably essential skills for being an effective physician.
The connection between Mayo Clinic and Luther is strong. We have in the Rochester community, so we have over 1,000 alumni and friends in that location, many of them at a large variety of positions at Mayo Clinic. I mean, we all think kind of primary healthcare delivery, but it includes the full swath of different areas of being a healthcare provider. And it also includes people behind the scenes doing the lab tests, the diagnostics. It includes people doing the statistics. This morning we heard from one of our alums on a career panel that is doing some of the statistics work for Alzheimer's patients in clinical trials. And so, yes, we have, that connection is strong. The Rochester semester has been developed based on that. And there are some great opportunities there.
Well, I'd like to thank you for attending this webinar, and please feel free to contact us here at the Luther Biology Department if you do have any more questions, and let's everybody take care and stay healthy.